we can make multiple kind of modifications inside a DNA, where we can utilize this Cas9 protein to cut the DNA. We can also utilize this Cas9 protein to induce certain changes without inducing any DNA cut. So all these things we will discuss in this in today's class and maybe the subsequent class, which we will be taking for another 2 days. So can you guys see the screen now?
Yes, sir.
Yes, sir.
So in today's class, we will study how we can design a guide RNA. So for that, if you look at this structure here, this is a Cas9 guide RNA complex along with the target site in your interest of gene, where you have designed a guide RNA against a gene, where this guide RNA is going and binding along with this Cas9 protein, and it is cleaving the DNA strand, which is targeted by this specific guide RNA. Now, how we can design this guide RNA for a specific gene which we want to make change? So for that, there are multiple methods that we will be discussing in this class. But before that, we need to know what is a guide RNA here and why it is important to design a guide RNA against a specific Cas9 protein. So now, if you look at this structure, this guide RNA, this entire complex is composed of 3 different kind of part. The green part which is highlighted here is the CRRNA. Then there will be a linker group. If you can see this purple region, this is a linker region that is basically joining this CRRNA along with the scaffold tracer RNA, which is highlighted in the blue color. So when we talk about a guide RNA, this will be composed of 3 different parts, and this entirely, when it is combined, then we will say it as a guide RNA. However, when we are talking in context of a DNA targeting part, then the CRRNA is the major RNA that is actually binding towards a target region of a gene. So when we are also designing a CRISPR Cas9 specific guide RNA, the major focus will be to design a CRRNA. The remaining part, which is this linker and the blue region, it will be constant throughout. And that is why there are different kind of methods where what we can do, either we can design a guide, we can use a certain system where the guide RNA, when we are designing, we will design it by using a certain vector where this guide RNA along with this linker group and scaffold